Hello and welcome to the fourth episode of the SERA Space Collective. I am your host, Sam Hutchison, co-founder of the Space Exploration and Research Agency, or SERA for short. Today, my guest is Dr. Eliah Overbey, who is a pioneering researcher in the field of astronaut health, specializing in spaceflight-induced genomic changes. More on that later. Dr. Overbey is professor of bioastronautics at the University of Austin, Texas, and the chief scientific officer at BioAstra. Through her lab at UATX, Dr. Overbey is leading the design and implementation of SERA's own space science competition that will see 24 experiments flown on the Blue Origin New Shepard rocket on our upcoming mission. Today, we're going to be talking all things human and space. Thanks for tuning in, and please enjoy the show. Dr. Overbey.
Sam Hutchison.
So nice to see you. Thanks so much for getting on.
Thanks for having me on.
So, Dr. Overbey, I'm going to start you with a low ball, if not a slow ball. Did you always want to get into space exploration and research? Were you one of those children who was staring up at the stars and just counting down till you could get your NASA application in, or did you get there a more sort of roundabout way?
Yeah, so I got there a little bit of both. I was always fascinated by space. I remember seeing a movie when I was in middle school where people landed on Mars. It was a fictional movie. But when I was a kid, I thought that we had actually done that already. I knew the moon landing was real. So I thought if we were making movies about landing on Mars, we must have also landed on Mars. Turns out we hadn't. Mars is much different than the moon. But I didn't get into space right away. I studied computer science. I interned and worked at some tech companies for a while. I ended up going to grad school and studying genomics, and that's where I ended up getting interested in spaceflight. When I was in grad school, there was an astronaut, Dr. Kate Rubins, who was doing research on the International Space Station. She actually did the first DNA sequencing experiment in space, and that got me thinking, how many times have we done this before? Have we done anything like this before? What do we know about astronauts once they return to Earth? What are the genomic changes they undergo? And we hadn't collected that data. This was the first time we'd sequenced DNA in space. We hadn't done any more complicated omic sequencing or methodology up there. Not only that, we weren't really studying astronauts' different omics profiles once they return to Earth. So we weren't looking for mutations caused by radiation or changes in gene expression levels.
Why not? It seems pretty critical. So they were looking more at the kind of sort of physiological behavior of the human body in space as opposed to the changes it might be going on a cellular level longer term.
Exactly. Yeah. So ever since the space program started, there have been doctors looking at what changes in astronaut health during flight. And this is changes in muscle mass, changes in bone mass, vision changes. What started happening, late 90s, early 2000s, especially after the Human Genome Project came out in the early 2000s, was there's now this opportunity to start looking at different genomic changes in various diseases. However, there was quite a bit of lag between when that technology was available for studying something like cancer or Alzheimer's and then when it actually started getting applied to astronaut health. You'd asked about the reasons for this?
Yeah.
The reasons for this, there's a few reasons for this. One, omics research, the cost of it is going down, but it's still expensive compared to other ways of monitoring human health. NASA is primarily an engineering firm. They develop rockets, and the humans are typically just part of that payload that they're sending. And the reason behind this as well is that we don't have that many astronauts going to space. It's still a very early field. So there hasn't been that investment in it. Another reason is that omics research is very sensitive. It's about as personal as you can get about your own biology and physiology. And there are, concerns, I think, on NASA's side about that, what where you can distribute that data, how far you can let it go. So generally they keep any data they've done on genomics very close to the chest, to their internal researchers. The other reason being astronauts themselves aren't particularly keen to participate in this type of research because you can't change your genome. So if somebody finds something, if a scientist finds something that indicates you might have increased susceptibility to radiation damage, for example, there's nothing you can do to change that. And it's possible that you might be grounded and not prioritized for a mission because of something you can't change. So that's why one of the many reasons why private space exploration is so exciting, commercial space exploration, is because when you're paying for your seat, nobody's going to ground you. In fact, you might want that information to inform your own personal decisions about what types of space missions you want to personally undertake.
That's so fascinating. It's a classic instance of sociological and psychological reasons impeding good science, but it makes perfect sense. Is that attitude and culture changing? I know you've worked with a number of astronauts in the past. Are they predominantly private, or have you managed to unlock some of the National Space Agency astronauts who are up for longer duration flights?
Yeah, so my mentor, Chris Mason, he's had some luck with some NASA astronauts, particularly Scott and Mark Kelly for the NASA Twin Study. But ever since the NASA Twin Study, there haven't been these large public-facing genomics data sets that have been created. Even the genomics data sets from Mark and Scott Kelly aren't publicly available. We have much more luck through the commercial sector. So I've studied the Inspiration 4 crew, working on Polaris Dawn and From 2 right now. We've collected samples from Blue Origin astronauts, Axiom astronauts. So the commercial side has been much easier to work with in this regard.
And what should we be worried about if there hasn't been significant research on genomic change? What are the risks? I'm obviously sort of thinking about longer duration spaceflight, Mars missions. You mentioned at the start of the show. What is lurking out there that you're focused on, that we should be worried about, that might impede longer duration human spaceflight?
Yeah, one thing we're noticing, so we collect a variety of biospecimen samples from astronaut crews. One of these samples is blood. We also get urine, stool, saliva, skin swabs. But from the blood, what we can do is we can isolate plasma and look for circulating cell-free DNA and cell-free RNA. So what this allows us to look at is gene expression from cells that have lysed and is now circulating in the bloodstream. What we see with different proteins circulating in the bloodstream. Can I go back? Hang on.
Yeah, sure.
Okay, can you give me that question from the top?
Sure. Given the lack of historical research in this area, what are the issues that we should be worried about that might impede longer duration human spaceflight, Mars missions, and beyond?
Yeah. So we collect a lot of different samples during spaceflight: blood, urine, stool, saliva, skin swabs. But from the blood, what we can do is isolate plasma, and from there isolate proteins that are circulating in the bloodstream. What we've seen from those proteins circulating in the bloodstream is increased representation of proteins we typically find in the brain. One thing that we know happens during spaceflight is degradation of the blood-brain barrier. It becomes more porous. So what we could be seeing with these more abundant proteins circulating in the bloodstream is evidence that that blood-brain barrier is becoming more porous and potentially causing more crosstalk between your blood fluids and the rest of the body, your brain fluids and the rest of the body. And this is happening with short duration missions. It's happening with the Twin Study. The longest we've had a human in space for is about a year. That was how long Scott Kelly was up there for the NASA Twin Study. His record has been broken at this point, but not by very much. If we're looking at something like a Mars mission, which is going to be, if you're bringing people back, a minimum of about 2 years, you're doubling that amount of time in space. And we don't know if these issues that we're seeing are going to get exponentially worse, or if the body can adapt and sort of control for the issues that are arising.
And how many of the sort of areas of concern are related to radiation, and how many of them are related to the the actual experience of sustained microgravity?
Yeah, those two are hard to study in isolation. Certainly, when you're looking at radiation, you're looking a lot at DNA damage and different stresses, inflammatory stresses that radiation could cause. We have to take into consideration that the International Space Station is still protected by partially the Earth's atmosphere and partially the Van Allen belts. So the radiation doses you're getting on the ISS are not representative of what you're getting on the moon or on Mars or on any sort of deeper space mission. So the best way that we can actually simulate radiation, that type of radiation on Earth, are through particle accelerators, like the ones at Brookhaven National Labs. But even those are limited because you can't, if you want to do a study, you're not putting humans in those for starters. You could put something like a mouse in there, but since space on these machines is so limited and costly, you're not keeping them in there for very long. So we don't have great models for what happens to any sort of biological entity on the timescale of a Mars mission.
And are there any plans to do the sort of work that needs to be done as part of the Artemis missions?
Let's see, the Artemis missions. Are those happening?
No, I know. I'm talking. Fair point. But are you aware of any work that's going to be going on in terms of humans on the surface of the moon over the course of the next decade?
Yeah, so NASA has been soliciting scientific proposals for so they've done science on Artemis 1. So Artemis 1 was a mission around the moon that did not have any people on it, but it did have scientific payloads. And so there were organisms flown around the moon then. Artemis 2 will have humans aboard, but just for an orbit around the moon. Artemis 3 will actually put boots on the ground. There are project solicitations for Artemis 2 and 3. I don't know that they've selected the final rounds of science that will go on those or what that science will look like.
And I mean, based on the state of the art today, I mean, do you think that the scientific community has seriously underestimated the challenges to long-term human spaceflight? Or is just
the scientific community?
Yeah.
No, I don't think that. Actually, if you go to scientists, they're usually the ones that are most conservative about landing people on Mars, or the ones that are most cautious about it. That's where you'll see people trying to slow down our pace of exploration the most, because they want to understand every single thing that could possibly happen to the human body before we go there. It's this safetyism. If you go to the private sector, if you go to more of the normal human population, that's where you'll see more people wanting to take risks, and like actually go on these missions. So despite the fact that scientists don't have all of the data, I don't think it's really impacted their caution around spaceflight. They've erred on the side of caution rather than the side of the side of glory.
Sure. And in terms of the work that has been done for sort of long duration orbital human spaceflight, are we well set up to expand human activity and presence in orbit? Do you feel at this point? Is the data in? Are we good to stay longer? Are we good to send more people up? Have we understood fully, or are we still in the very, very early stages?
This is very early stages. Even for the experiments I've run, that I've conducted, if you're doing a study and you want to get a drug approved for market, say you need to have cohorts of at least hundreds of people that you're working with. Right. We don't have that. I mean, if you're looking at the number of people that have gone to space, the number of people that have gone to space in the entire history of human spaceflight, it's only about 650, 700 people. That's the range that we're in. When you look at the number of people in my studies, they're cohorts of between 1 to 4 people. That's not a lot of data. We know very, very little. We're at the very early stages.
Okay. And then in terms of the sort of space research ecosystem, is this research getting funded? Is this an area that has been historically ignored? I mean, I know all researchers and scientists are always looking for funding, but I mean, is it being underinvested in?
Hell yeah. Yes. Yeah, it's being underinvested in.
I mean, it sounds that way. That's what I'm asking.
Yeah.
But what can you actually do if you're always constrained by small sample size. Like, to your point, you've got 650 historical examples, the vast majority of whom have had no data whatsoever taken on them. You've got resistance from the sort of professional cadre of spacefarers. You've got some, let's say, braver souls who are paying their own way, and so who are willing to sort of give up their information for the good of the longer term human presence in space. But that's not going to get solved by anything. Are there simulations that you can do? Is artificial intelligence going to come into play? Are you going to be able to simulate, human bodies in space? And, are we at a point where there are economies of scale, or is this just a hard problem that can only get fixed by putting more people in space and having them be willing to share data relating to their genomics?
Yeah, I know a lot of people want to use AI to solve this problem. They say, well, we'll simulate the human body with AI, we'll find targets with AI, drug targets with AI. I mean, if it were so easy to do all of that, then we would have solved actual diseases here on Earth already using those methods.
Right.
It's not easy to do that, and we don't have perfect simulations for how the human body changes in microgravity. Even if you're just looking at changes in how fluids are behaving, that's a very complicated problem. People have studied that in isolation, looking at how fluids change in fuel tanks during spaceflight, and they've seen unexpected and unanticipated fluid behavior in flight compared to what their models were predicting. And the human body is just a very complicated water bag with all of these other things, all these other interactions in the way of fluid movement. There's no good simulation that we have for that. What we need is some way, until we can start sending people into orbit and scale and conducting clinical trials, maybe with something like Starship or some other high throughput way of actually getting larger cohorts of people in there. I don't think we're going to have satisfactory answers.
That brings me on to the project that we've been working on the last year, which is designing experiments to be taken aboard Blue Origin's New Shepard rocket. Is there a role that suborbital space operations can play? Perfect. How?
Yeah. So for the work that I do on the human body, for example, we're collecting samples from crews that have been up there for 3 days, for 5 days, for a couple of weeks, through a lot of SpaceX and Axiom opportunities. What we don't know, the sort of problem that we have, is establishing good control groups for those cohorts. One thing those cohorts will experience is the process of launch and landing. We don't have any way to control for that with terrestrial research subjects. So New Shepard astronauts, they don't get a whole lot of time in flight. They're just above the Kármán line. They're not getting a whole lot of radiation or microgravity exposure, but they do get that entire launch and landing procedure. So getting samples from these crews, and it's a lot easier, I think, to get high quality samples from Blue Origin crews as well for some logistical reasons. We can get really good control data from Blue Origin groups.
And in terms of the science competition that we've been working on, tell our audience a little bit about, I think both, you mentioned fluid dynamics. We've got a fluid dynamics experiment, and we've got a cellular biology experiment. Can you walk us through how experiments designed for suborbit can play a role in feeding into the sort of longer term orbital research that you're talking about?
Yeah, so one of the competitions we have for the SERA mission is a fluid dynamics competition, like you said.
Yep.
What we're looking for there is how fluids are behaving in the ways that we do molecular biology experiments in space. So we only have about 3.5 minutes to do an experiment on the Blue Origin mission. That's how much microgravity time you get once you subtract the launch and landing time.
Sure.
So we need to do something quickly in order to look at fluid changes in spaceflight. One issue in my field that we've had with conducting research in space is that a lot of molecular biology, so a lot of the ways that you isolate DNA, RNA, and process it for sequencing, involves manipulation of fluids. And when you're opening up tubes and moving them around the capsule in space, fluid flies out of them. You don't have that gravitational vector keeping fluid at the bottom of the tube. One thing that would be really helpful for space flight scientists would be to have a tube that could actually keep fluid in it. So what we're doing with the competition is we've invited people to submit proposals to redesign the 1.5 ml microcentrifuge tube. This is like the standard tube, that people use in laboratories, and redesign that to better hold the fluid in the tube. And what we have on our end is we have a Form 4 3D printer. We're going to 3D print the designs of the different competitors, and the winning competitor will have that design sent to space on the New Shepard flight.
And that's going to be performed by the astronauts that we're choosing from our community, correct? Are they going to have enough time?
They will have enough time, yes. So we've designed a few simple movements to take the tube through. So what we have is that each crew member will have a GoPro or some sort of filming device attached to them so that we can see what they see. What they'll do is they'll have three tubes of that winning design. So each seat will have a different winning design that was selected from whatever country that seat has been allocated to. One of the tubes will be filled about a third of the way up, 2/3, and then the last one will be filled all the way up. So we'll get to see, they'll uncap the tube, they'll go through a series of motions. I'm trying to hold my hand up here on the screen and not get it edited out by the background I have, but they'll go through a series of motions, first side to side, and then up and down. And we'll be able to see, did this tube actually contain the fluid, or did it escape? And for when you only have a third or maybe even 2/3 of the tube filled up, that might not be a very difficult thing to achieve. But if you have a tube filled all the way up, that's going to be much more complicated, much more challenging.
I mean, what I love about this experiment is that it's demonstrating ways in which suborbital research can contribute directly to the design of equipment that could be used, for example, in orbital research, right? It's a no-brainer. The other experiment that we have going on is more complex and more in line with your specific field of interest, and it's around human cell behavior in microgravity. Tell us a little bit more about how that's going to work.
The other competition that we have is a cellular biology competition. So for this one, we're actually sending cell cultures to space with the crew. This will be located underneath their seats. So it'll be an autonomous payload. The crew members themselves won't have to touch or access any of it. It'll be operating independently in the background. And in each of these boxes, we'll have a few different cell lines that are being flown. And the competitors get to pitch which cell lines are selected and which morphological markers they want to see stained. So what we're looking at here is, I'd mentioned earlier, the human body is basically just a water bag with all this complicated proteins in the way, restricting its movement. And so what that is in practice are all the different cells in your body. And when you go into space, those cells are going to have different fluid pressures on them based on those fluid shifts. And that can cause changes in behavior of those cells. So what we're going to see here is we're looking at all these different cell types. Each seat will have a few different cell lines flown underneath it. So we'll get about 18 cell lines in total that we're testing. We'll stain those cells once they get back down to Earth with different morphological markers to see how is the morphology of these cells changing in microgravity compared to the human body on Earth, or these cell cultures, our control cell cultures on Earth.
What can you expect to learn? I mean, what sort of changes can you anticipate? What would be a win? What does a win look like on one of these experiments?
Yeah, so one of the things we'll be looking for, we'll want to hold some markers consistent across all cell cultures. And so what we'll look for there are different changes in the cytoskeleton. So we'll look very precisely to see reorganization of cells. And once we see that sort of reorganization and structure, we can go back and we can look at different diseases on Earth that have similar restructuring to try to predict what effect spaceflight might have on the body if we see this effect happening for longer durations.
And I mean, again, speaking from a position of almost pure ignorance, can you see changes in such a short period of time? It's,
Yeah, so there have been some, what's that?
It's immediate. It's as soon as they're in microgravity, you're going to start to see the shift, and the shift that you see in a short duration of time is contiguous with what you would expect to see over a long period of time.
Yeah, so you can see changes in a short duration of time. There have been some experiments on sounding rockets that have seen this. Those
Sure.
are rockets that essentially do the Blue Origin trajectory or something very similar to it, just without humans aboard. And there haven't been very many examples of that done, but it has been done before and they have seen differences. Um, the hope is that if we can see what's happening over a short period of time, we can map it to diseases back on Earth and look how those diseases progress, to design potential countermeasures for if astronauts experience those same changes over extended periods during their missions to the moon or Mars.
Understood. I'm super, super excited about it. The last thing that I know you've got to go. I asked you about challenges in scientific research. I know that you've obviously relatively recently joined the University of Austin, Texas, which is in the news a lot recently. How is UATX different to other institutions that you've worked with? What remit have they given you? How are you ripping up the rule book and doing things differently when it comes to space research at UATX?
Yeah, one of the things that attracted me to UATX is they have a very strong mission towards entrepreneurship,
Yeah.
which one of the things that has been frustrating about space travel is that, you know, we've done some very ambitious things in the past, but we haven't done them again. For example, we went to the moon, but it was basically a camping trip. We haven't been there in about 50 years at this point.
Yeah.
And the issue with that is not, you know, technological, it's economical. It's how do we sustain these expensive missions, or how do we bring the cost down so that we can do more of them? And so when you're thinking about space, you have to be thinking, how can I make this cheaper and how can I make money off of this? How can we make this a part of the economy? So the idea of having students that are very entrepreneurially oriented could prime them to be very big movers and shakers in this emerging economy, space 2.0.
Is it fair to say that the sort of traditional model when it comes to space science has been work out how to get those research grants, get those NASA applications in, get the federal dime, and off you go? Which, by the way, is an extremely lengthy, frustrating, often bureaucratic process.
Yeah,
So the idea with UATX is get people off that sort of government research funding track and get them thinking about how they can, build businesses around the research that they're interested in.
exactly. It's not to say there's no role for government funding. I mean, the
Sure.
government gives out a lot of funding. If you can get those grants, that's great.
Sure.
But if you're thinking about sustainable spaceflight, if you're thinking about, you know, actually starting a company, getting people to the moon, getting people to Mars, and keeping them there, keeping them alive, starting economies, starting, you know, new governments from scratch. That was another thing that attracted me to UATX, was they think a lot about like history of philosophy, a lot about the great books, how civilizations start, political theory. And as I'm thinking about, you know, after we get people to somewhere like Mars and they don't have immediate communication with Earth, they have
Sure.
to, you know, get along with each other and start a new civilization essentially from scratch. What sort of people, what do we need to teach those people in order
Sure.
for that project to be successful? Because we saw that happen when people were colonizing the Americas, because there were a lot of failed endeavors there.
Sure.
And how do we minimize the number of failures that we see this next time around?
Well, I hope it's sooner rather than later. Dr. Overbey, thank you so much for your time. I think we'll be releasing more details about how people can get involved, submit proposals for the science experiment competition next month. And then we'll be kicking off. You'll be a fantastic resource for the community as they put their ideas on paper and try and get those cell cultures and fluid dynamics experiments in space. Thank you for your time, and I look forward to seeing you soon.
Yeah, thank you for having me here. I'm really looking forward to kicking this off and seeing what sort of submissions we get.
Yeah. Thanks so much, Dr. Overbey.
Okay, thank you.